what I'm going to do first is briefly summarize some of the work that our group and our collaborators have been carrying on to try to figure out the physiological roles of insulin-degrading enzyme, as well to provide some of the first ever proof of principle experiments that validated this target for type 2 diabetes. So to kick off, these panels represent the main strategies that we have nowadays to treat diabetes. And they hinge on some aspect of insulin biology to try to lower blood glucose levels, which are toxic when elevated and also cause all the undesirable symptoms of diabetes when left unchecked. And the oldest method by far is the direct injection of insulin supplements with these insulin analogs that have extended half-lives. We can also force the pancreas to produce a little bit more insulin with small molecules and these hormone mimetics. And we can also sensitize the cells to the action of insulin to some extent. However, there is a fourth approach that up to now has not been realized into human therapeutics. And we believe that inhibiting the degradation of insulin that has been produced by the pancreas endogenously in response to nutrients, it's a very logical and complementary way to lower blood glucose levels. And because this approach is compatible with some of the pill-based small molecule treatments that are shown on this slide, we may be able to find a unique treatment combination that spares patients of the daily and weekly injections that they need to take for insulin supplementation. So there seems to be a lot of drugs in this field, but there's still a significantly unmet medical need to properly control blood glucose. So we're missing an image here. See? Okay. So there's a nice picture of insulin-degrading enzyme on the right-hand side. There'll be others anyway. So insulin is an extremely dynamic hormone. Its half-life in vivo is just under 5 minutes. And the main pathway of inactivation is by proteolytic cleavage by this very aptly named enzyme. And IDE is a zinc-dependent metalloprotease that is ubiquitously expressed. And it's actually one of the few protease genes that are linked to the disease by genome-wide association studies in human populations. So it is a good candidate for this job of inactivating insulin. But even well before this modern era of genetics, we knew about this enzyme because it has been isolated as far back as 1949 by careful fractionation of cow livers. And then it was cloned in the 80s and further characterized in the 90s. So there was decades of speculation that perhaps blocking this enzyme in an animal may lead to a treatment for type 2 diabetes. But up to now, it has not been possible to validate these hypotheses because we lacked inhibitors that were both specific for this enzyme and active in vivo. So we entered this field when we discovered the first physiologically active inhibitor of IDE, starting from an unbiased DNA-encoded library of macrocyclic peptides. And in this approach, the entire library of DNA-encoded compounds is exposed to the target in a one-pot experiment. And the target is immobilized on beads using an affinity tag. And that generates an enrichment perturbation in the composition of the library that you can deconvolute by high-throughput DNA sequencing of all the DNA barcodes. And when we decoded the barcodes that were enriched by IDE, as you can see on the left, we decoded the structures and synthesized them in large scale. We found that these were reasonably potent inhibitors for the enzyme, showing interesting SAR, confirming the interaction. So next, we investigated these macrocycles by replacing each building block and validating biochemically, but to cut a long story short, we arrived at this compound that had very good properties as the first physiologically active probe. And we also created this inactive control compound that is identical in connectivity, but it has a couple of chiral centers switched so that it abrogates binding to the target. And that will be an injection control in some experiments. One serendipitous observation that was really helpful for the development of these compounds, as you can see in these IC50 curves, is that these macrocycles were exquisitely selective for IDE, well over 1,000-fold versus all these other representative metalloproteases from other gene families. In particular, when you compare to this more traditional inhibitor that is not very selective because it relies on an interaction with the zinc atom in order to inhibit the enzyme. And these enzymes don't have very different catalytic pockets, so it's not very selective. So I'm going to skip the pharmacokinetic study, but suffice it to say that with one injection of 6-PK, we can engage the target for around 4 hours at 100-fold or more the necessary concentration for inhibition. So during this time frame, what we can do is perform some of the standard experiments in the field, such as the glucose tolerance test that is the same assay that is used to diagnose diabetes in the clinic. And in a typical experiment, you'll see the surge in blood glucose levels that then controlled back down to baseline at a rate that depends on the metabolism and the insulin resistance of the animal. So this is the result of the same procedure run on a diet-induced obese mouse that has been fed a high-fat diet throughout its life in order to elicit insulin resistance, which is a model of type 2 diabetes. So in the key experiment, when these mice were treated with the active compound here in green, they displayed significantly faster clearance of blood glucose compared to the animals treated with vehicle or the inactive control in red and black. And this also worked with the lean mice and the diet-induced obese mice. And this data represented the first observation and the first demonstration that acute inhibition of insulin-degrading enzyme leads to an improvement in glucose tolerance by boosting insulin levels. And on another set of experiments, we showed that the effect of IDE inhibition is actually quite comparable to this FDA-approved drug, Januvia, that was mentioned before. And this drug works on a completely different enzyme, on a different pathway that increases insulin secretion. So these two strategies can actually be combined to have an additive effect. And we think that this is one of the very interesting combinations that may enable us to spare patients of insulin injections before meals. So, however, when we were doing more mechanistic experiments, we decided to repeat this experiment, but in delivering glucose by injection, which basically bypasses a regulatory layer of hormones between the gut and the pancreas. And what we saw was this striking phenotype. The only difference between these two experiments is the delivery of glucose. And we saw this striking phenotype that suggested that we were missing something in our model. And when we measured the hormones at these early and late time points, we saw we could assign this phenotype to elevated levels of glucagon, which is the hormone that physiologically does the opposite action to insulin. So this observation then led us to a number of biochemical and physiological experiments that demonstrated that IDE really modulates the three pancreatic hormones, insulin and glucagon being the main yin and yang of glucose control in the body, as well as this secondary hormone amylin that is co-produced and co-secreted with insulin by the alpha cells. And in the interest of time, I won't show you the endogenous physiological signaling experiments, but instead just the direct injection of each of the three substrates and the effect that IDE inhibition has on the blood glucose effects as well as the plasma levels. So to recap up to now, using the first physiologically active compound, we could demonstrate that we can improve glucose tolerance by IDE inhibition. However, this mostly relies on a moment in which the pancreas is responding or coordinating a response to an intake of a nutrient, like a meal. And at this point, there is really high secretion of insulin and suppressed secretion of glucagon. But in conditions that do favor some glucagon signaling, we can see elevated gluconeogenesis. And this is still very promising, especially if you consider all the FDA-approved drugs that we can combine with these inhibitors. But it is still a potential hindrance to the translation of these compounds toward the clinic and the daily routine of patients. So we asked this question, what if instead of using this blunt force inhibition approach, there was a more of a velvet glove approach to modulating insulin-degrading enzyme in a way that it favors the substrate that we want? And hopefully this video will illustrate why we thought that we had a reasonable chance of succeeding, even though it is a hypothetically impossible feat for most enzymes. So here you see 6-PK bound to the enzyme and is binding within the cavity in a distal site, far away from the catalytic center where the zinc atom is, around 11 ounces away. And by sitting in this pocket, most of the macrocycle is occupying the cavity, and this volume will obstruct the binding of a substrate that will use that wall in order to be recognized as a substrate. So we asked the question, what if we could modify or find second-generation compounds that might be able to discriminate between insulin and glucagon based on their differences and the ways that they bind insulin-degrading enzyme. So instead of, so this will be full of dots showing hits, but I'll just skip over and I'll say this. Instead of trying to modify our first-generation inhibitor, what we did is to refurbish it into a fluorescent polarization displacement probe shown on the bottom. And what this does, it reveals a drop in polarization signal whenever it gets displaced out of the IDE cavity. So the plan was to screen 100,000 compounds from the diversity-oriented synthesis library at the Broad. And we actually stopped around in a quarter of those hits because we found a sub-library that contained 100 hits that were displacers. And from those 100 hits, I could find a handful of compounds that show this discrimination property between the two substrates. And these are dual antibody assays for insulin in green and glucagon in red. And this happens over a range of concentration, unlike 6PK that shows an identical EC50 for both of these substrates, as well as an 100% inhibition maximum. And I apologize that now on these structures will have to be redacted for IP purposes, but you can see that these compounds are now significantly smaller and drug-like and very promising for pursuing them. Another cool feature of the way that we performed that screen is that these new inhibitors now inherited the excellent specificity of 6PK because they bind that distal side pocket that really doesn't exist in any other metalloprotease. In collaboration with the Siliger lab, we recently obtained a co-crystal structure of one of these analogs, and we validated with mutations in those sites shown there. And we can now show you how we think they work. If we superimpose the IDE insulin co-crystal structure, there is a major steric clash with part of the molecule sticking out into the IDE cavity, as shown there. But glucagon, because it is smaller and more flexible, it can still use the same wall of the enzyme to interact with IDE on two regions, but it can engage now the catalytic site in a biochemically productive way. So depending on the assay and the compound that we're using, we see between 60 and 80% of the native activity of the enzyme. And just to wrap up, one huge drawback of rescreening is that now you have to re-optimize your compounds again. And these compounds were around 5-fold less potent than the first-generation inhibitor. But we hit some inspiration when we realized that these compounds might be paying an entropic penalty by binding the target, having to flip these rings out of the plane. So when we locked them in this conformation, we got what's called a magic methyl effect that improved affinity by 120-fold. And now you can see that we have these very nice molecules with high potency, and they discriminate between insulin and glucagon in over 3 orders of magnitude. So what we can do now with this new generation of inhibitors is really block inhibition of insulin degradation over long periods of time, allowing glucagon to get degraded. And we can see also in the mass spec it going away, and the main cleavage site is still the same one as the native enzyme. So that is perfect for going forward. So I hope I've convinced you that IDE is a compelling target for the treatment of type 2 diabetes, and that because of its in vivo functions, we have to be a little bit smarter about how we go about inhibiting this enzyme. But now that we've discovered the substrate selecting inhibitors that bind the 6PK site, we can now selectively disrupt the insulin interaction, allowing the glucagon interaction to continue. And just a quick shout out to my mentors and some of the collaborators and colleagues that have helped with the story, with the validation story with 6PK, as well as the Broad Institute collaborators that helped us put in place the screening assay that turned out to identify these second-generation compounds. And I should say that looking forward, what we hope to do is translate some of these findings into other biomedical targets of interest that have this polyspecificity problem, for which we are currently, they're out of the scope of current inhibition approaches, or for which we are asking patients to put up with some sort of undesirable side effect from their medicines. Thank you.